Hello, students. I hope you're doing well. I'm sorry I'm a bit behind. Technically under 24 hours, but I've had strep throat and I have an eye infection, and there's no good excuse in academia. But I appreciate your patience. This is a 2-week lab, so we don't have a deadline coming up anytime soon. Know that there are 5 parts to this lab. 4 of the 5 parts can travel, so you can plan ahead, do different components. If you're on the road over the weekend or something, you don't need a lot of time at your lab bench at home. The one component where we extract DNA, that's about 30 minutes, and you will use supplies from your kit for that part of the lab. So in the first lab section, as you know, I've been working closely with my colleague, Dr. Hollis Brown, to make sure that your online lab experience is very similar to our classroom lab experience. She teaches on Wednesday, I teach on Thursday. If you ever want to visit us, you're welcome. So you will go through the reading material and you're going to answer questions wherever you see arrows. So recall that you can submit just the arrowed parts of these labs, or you can submit the whole document, whatever is best for you. How do I know how many points to deduct per arrow? I actually do a control find after I write the lab and I look for the arrows. So in this lab, there's like 37 arrows. So I will be deducting 1 point per question missed. And so as you can see, 2 weeks. This 2 weeks in the classroom lab is 6 hours. So this is a long document and I hope you take it in stride. So after reviewing the lecture materials about DNA and how we go from DNA to gene expression, you will do 4 components to this lab. So on page 11, we'll start the online lab. And so about 1/3 of the students in 1060 are criminal justice related degree programs. So I've been looking to find a lab for this course that really focuses on this career application and this degree programming. So in the first section, we're going to do a forensic biology exploration. In part 1, you're going to read about next generation forensic biology. This is an interesting article written by PhD students at Harvard University. As you're reading, you can take this 5-question quiz about the reading. This is just my way to show you how your assessment relates to your study tactics. And so work here until you get all 5 points. You can take the quiz as many times as you want. Take a screenshot of your quiz when you're done. That's what mine looks like. And then I'll know you did it because your email will be here and I won't have to go look for every single person's quiz. Thank you very much. In part 2, we're going to look at this illustrated guide to home forensic science experiments. This is like a several 100-page document about all the cool things you could do at home to model the kinds of practical applications that exist in forensic biology. Of course, I don't expect you to read the manual. Just go to the table of contents and click away at some of the experiments that might interest you. And then choose one. There's all sorts of stuff, from splatter identification to soil science to hair analysis, all sorts of stuff. Paint analysis. So just choose an experiment. And then you're going to read that experiment and then digest that, right? 200 words about the background information, that's a section header in the labs. And then 200 words about the procedures, another section header in the labs. Finally, type the review questions for that lab here and then answer them. They might ask you about experimentation, but I spent quite a while reviewing it, and most of them were about that background information. And then finally, just tell me about yourself. Are you a criminal justice major? Was this interesting to you? Because this is a new component to my labs. Part 3, DNA extraction. So this is the fun manipulative part. You can do a virtual DNA extraction at the Genetic Science Learning Center. This will show you how it's actually done in the lab, but guess what? We can do that right in our homes or dorm rooms as well. So there are two procedures I've chosen here, one from the University of Utah Genetics Learning Center, and then Science Buddies, another one written by Scientific American, an academic popular media source. Here are the materials you will need to round up. A lot of the random chemicals that people decided not to take from me would be employed here. If you decide that you do want to get some soap or salt or soap or rubbing alcohol from me, just email me and we'll figure that out. Then tell us which DNA extraction lab you chose. Tell us why we used all those chemicals to extract the DNA. Take a picture of you and your DNA extraction setup, and your index card with the date and your name. And then finally, in the last section of this lab, you're going to explore a biotechnology application. And we're going to couch this in a conversation about sustainability. So here's the sustainability Venn diagram. And this is a common tool used to kind of ascertain whether or not economic, ecological, social activities are sustainable. Do they reflect these basic characteristics of our global community, the environment, our economics, and our social well-being? So you'll define sustainability, describe the diagram in your own words, spend 30 minutes doing research, looking up biotechnology applications. There's all sorts of them. You might have heard that there are fish genes in tomatoes. You might have heard that they can put um, chemicals right into vegetation that fight herbivory from insects. You might have heard that you can put nutrition in rice. So there are lots of ways that we've used biotechnology in our society in the last 20 to 30 years, and I'd like you to just explore them freely for yourself here. We're paying close attention to the quality of our references, so please cite 3 references here and tell us why you think they're a quality reference. And then finally, put that into the reflection on sustainability. I've heard some really amazing arguments from students over the years for and against the use of biotechnology. It's really not a straightforward conversation. There are many different stakeholders and facets that one needs to consider when considering whether or not we should support biotechnology in our modern society at large or just for research purposes or some other qualification that you might dream up. In the end, as always, write 100 words of reflection so I know how to improve this lab and what the strengths are that I should retain. And if you, for example, choose to skip over the DNA extraction lab because you're going to come and use the molecular model, you might not have the photo, so I wrote it again here. You don't need to copy the photo of the DNA extraction lab twice, but if you don't do it, here you might put some other photo. Okay, I hope that's all clear, and please let me know if you have any questions. I have extended that discussion deadline to the second Tuesday, but I urge you to take action right away. And I will be very busy grading your photosynthesis and cell division labs this week. Thank you very much.